The Insight.
Insight.
The Insitome.
Podcast.
Genetics.
Human.
Archaeology.
Denisovans.
Neanderthals.
Metabolism.
Ancestry.
Where in the world did we come from?
I am.
Unique DNA.
Genome. From Austin,
Texas.
This week, consumer genomics goes mainstream. I am Spencer Wiles, founder and CEO of Insitome, and
Razib Khan, Director of Scientific Content at Insitome.
And Gareth Heinum, Director of Genomics at Insitome.
and we're going to talk about the development of the consumer genomics industry over the last 15 years or so. So we're in early 2018 now. And the industry does date back about a decade and a half. Back in the early days, so this was kind of 2002, 2003, there were two early players. It was very much a cottage industry, like the most cottagey of cottage industries, I've called it.
Yeah.
Really kind of people who had heard about scientific papers that had been published, or they'd read popular accounts. Brian Sykes, who was a professor at Oxford University, wrote a book called The Seven Daughters of Eve about the founding mitochondrial lineages in Europe. And a lot of people were curious after reading the book, hey, where do I fall? Do I belong to one of these seven? And can I learn something about my ancestry by testing myself? And so he started a small cottagey company out of his academic lab to allow people to find out where they fell into one of those seven families. Mike Hammer at the University of Arizona had been involved in a study on the Jewish priests at around the same time. And he and Bennett Greenspan, who went on to found Family Tree DNA, started offering people the opportunity to see if they fell into one of these Cohen modal haplotypes and if they were therefore a descendant of Aaron and part of this priestly caste, if you will, within Judaism. But yeah, I mean, literally these companies at that time were testing maybe a dozen or a couple of dozen people a month. It was around that time that I was finishing up Journey of Man, the film and the book, and we're actually going to be doing a Journey of Man Redux podcast episode. And as part of the promotion for the film, we actually did some testing, some Y chromosome testing on a few celebrities around the world. So this was for the international release, which was on National Geographic Channel. And so that came out in December of 2002. So kind of late summer of 2002, fall of 2002. We were collecting samples from people around the world, people like Jackie Chan in Hong Kong.
Very cool.
Yeah, it was kind of cool. And everybody was like really interested in getting their results. And so, when Journey of Man and my involvement with Nat Geo started to morph into the Genographic project the following year, I was like, we need to be doing genetic testing on people. And so, looking around at what was available at the time and realizing that, in all likelihood with National Geographic behind it, it was going to be a significant number of people, a lot more than a couple of dozen a month. We started thinking, like, who can handle the capacity? And we ended up deciding to work with Family Tree DNA. And IBM, our partner in Genographic, kind of helped them to scale their lab, working with Mike Hammer's group at Arizona. And so, we launched that project in April of 2005 and sold 10,000 kits in the first day, which I think is like more than had been sold altogether by anybody working in the space up to that time.
Quite a lot for 2005. Yep.
Yeah. And let's step back just one moment and remember that the draft of the first human genome had just come out in the year 2000. So when we're talking about personal genomics, these are really, really early times. Yeah,
and in those early days, we were not doing genome-wide analyses.
No.
We were focused on these uniparental markers, the mitochondrial DNA and the Y chromosome. The reason Journey of Man was called Journey of Man is because it was based on Y chromosome markers that we had discovered at Stanford and Luca's group, particularly Peter Underhill and Peter Uffner in that group, two postdocs at the time. And so, the early tests were all focused on just those uniparental markers.
Yeah.
They were much simpler than the stuff that's available today with hundreds of thousands of markers scattered around the genome.
So how many markers, I know some of them are microsatellites, which is a somewhat different thing, but how many markers were these early tests doing? Well,
so we basically improved a little bit on what Family Tree DNA were doing at the time, which was 12 microsatellites, or short tandem repeats, STRs. So these are segments of DNA that have repeat units like CAG, repeated X number of times. Yeah. And X can vary between, say, 8 and 18. And you tend to add and subtract entire subunits. And so, you end up with a distribution of types. They're highly variable. They mutate at a very high rate. And so that allows you to peg someone quite well in forensic uses or for ancestry testing to see if you're actually closely related to somebody on the Y chromosome. It's a little bit harder to infer the ancestral story on this, what we call the haplogroup. So this is kind of the ancestral clan.
Yeah.
But you can do it with enough data. And so
Yeah.
we started building up that data. We had to do some SNP genotyping to assign some people to haplogroups. But for the common ones, R1b in Western Europe, which is like 70% of men, it's very easy to pick that out on the basis of STR haplotypes. And of course, we know the reason why now. It's because they all coalesce back to basically the Bronze Age, just a few thousand years.
Yeah. So they're all very similar to each other. And with STRs, I mean, my personal experience with STRs is the high mutation rate. It's good because it causes a lot of variation, and it's bad because the mutation rate is so high. To calibrate certain analyses is a little difficult sometimes. So it's kind of a work of art.
Yep.
I think in some ways.
From next gen data, I mean, genotyping STRs is still a method that's in the works today.
Yeah.
I actually did part of my thesis around 2010 on genotyping STRs.
Okay.
So next gen meaning next generation sequencing.
data.
Yep.
Yeah. And so, Spencer, you have been talking mostly about, let's say, 2000 to 2005 or so, and we are in the cottage industry stage.
Well, so, yeah, I mean, we sold 10,000 on the first day.
Yeah.
And we hit 100,000 in sales by the end of that year. So within about 8 months, end of 2005. And a couple of years later, 23andMe comes along. And Navigenics, which I was actually involved in as an advisor, scientific advisor. And, they came along and they started doing whole genome analyses.
Genome-wide.
Using some of the early chip technologies, which were very expensive.
Yes.
And so when 23andMe launched, and people, I don't think, would believe this today, but, those of us who were there remember, it was $1,000, $999 to get your genome analyzed. Navigenics was $2,500. So, didn't have a huge impact at the consumer level in terms of sales. And then the price started to come down a couple of years later. And I think Illumina, which made the underlying technology, the chips that were being used by these companies, started to realize around that time that, wow, there are hundreds of thousands of people a year buying these things.
Yeah.
This could be a substantial market. And so they started to reduce that price and make deals with the companies and so on. And so we started to hit on that sweet spot, which is, a little under $100 for one of these tests.
Yep.
Yeah. Well, I mean, so the original chip, do you remember off the top of your head, the original 23andMe chip was 50,000 markers, right? Something like that?
I don't remember, actually.
Yeah.
Yeah, it could have been.
Because I think when it got popular, it was a 650,000 marker chip.
Yeah, I mean, ultimately the platform
565.
that everybody seemed to settle on was kind of the Omni Express.
Yes.
Which was roughly 700,000 SNPs, at least the last iteration.
Yeah, and I have like 950,000 markers because I upgraded my 23andMe, et cetera, et cetera. So it kind of maxed out at 1 million, and for the purposes of what they were doing, really that's all they needed. But so we've gone from the year 2000 when we had the first human genome, and there were a few cottage labs a couple of years later that were doing a dozen markers on microsatellites. And then 2010, we have 23andMe with, almost 1 million markers, over 500,000 markers. And the price
is dropping. And, if you look at the adoption curves, the number of kits that were sold between Genographic, 23andMe, Family Tree DNA, Ancestry launched their product in 2012. Probably around 1 million, I've estimated, tests were sold by that point, so by the end of 2012. So over the course of a decade, since the very beginnings of this cottage industry, up through Genographic, the launch of 23andMe, and eventually the launch of Ancestry, it took a decade for 1 million people to buy tests.
Yeah.
And then the curve really took off.
Yes.
Then we saw the hockey stick after that. So
Yes.
starting 2013, 2014, everything goes exponential. And literally, last year, 2017, Ancestry alone probably sold like 4 or 5 million kits.
Was that a critical mass thing in the genealogy community, or was it like a consumer price thing around 2013?
I think price might have had something to do with it, but I think a lot of it was just, you hit a critical mass of people who had been tested. And so much of the reason why people are choosing to do this is because of word of mouth. You know, a friend of theirs has tested, they found out something really cool, you know, maybe about their ancestry, or they found some fifth cousin online. And, you know, you start hearing about it at a party. When you have 1 million people testing, you know, most of these are upper middle class Americans. You know, it's quite likely in a group of 40 or 50 similar people, you're going to have someone in that room at that point who would have tested, and they're talking about it. And so I think like this word of mouth viral spread really started to kick off around that time. So, you know, price had a little to do with it, and greater utility. So as more people test, it's more likely that you're going to find one of these matches, one of these genealogical matches in the database.
Yeah.
The family finding matches, a killer app.
Yeah, it's one of several.
Well, so you've done some work in this space, Gareth.
So I actually had the opportunity to work at Family Tree DNA as an R&D scientist. And when I was there, one of my big projects was called the Big Y. And so, like Spencer was saying, when this whole thing got started, they were doing lineage SNPs across the Y. But the Big Y was this sequencing of as much non-recombinant Y chromosome as we could possibly do, which allowed not only testing of the 70,000 or so markers that existed, but it was actually like 12 million Y positions that could give you these paternal lineage SNPs unique literally to your own paternal family.
Yeah.
So rewinding and thinking about like starting off with 12 STRs, and we're thinking about very broad haplogroups. Are you R1b? Are you R1a? I think, in fact, we have an R1a in the house.
Yeah, we do have an R1A.
We're 2 R1bs and 1 R1A here.
Well, I mean, you know, 1 R1A equals 10 R1Bs, so I'm fine.
So these are 2 of the major lineages in Europe and across Eurasia, in fact. But everybody falls into one of these haplogroups, and people have like a sense of group identity from this, you know, especially R1As. Apparently.
Yeah, Clam McDonald, represent.
Well, there's a bunch of history in the lineages, especially when you start looking at those geographical migration patterns. And then you also have, you know, the identification of famous historical figures in the lineages. Then you can relate to those guys. I mean, that's really cool.
Yeah, and so the idea with the Big Y was you were really kind of drilling in on very specific sublineages. So it was way beyond like, am I R1b or not? It's, am I like in this particular surname group?
Yes.
Like all the Welleses have the same lineage if you look at literally thousands of genetic markers. And so that's really cool.
Yep.
You know, to get
Most
that
of the
level
Welleses. That's why you use it at
That's
this sort of
why you use it.
granularity.
Well, no, that brings up a really good point. I mean, the Y chromosome should roughly track surnames, at least in European populations, because you tend to get your surname from your father in the same way that if you're a male, you got your Y chromosome from your father. That is not the case for a lot of the common names. So the name Smith doesn't have any particular Y chromosome signal.
That's true.
But if you have a relatively uncommon surname, and it was actually Brian Sykes, again, who did this study for the first time, who, you know, looked at his surname and he found a particular Y chromosome lineage that was associated with the majority of people that were named Sykes. And so, you know, you can see that kind of echo of the cultural effects in the genome.
Yeah. And that's why the people that are into the Big Y, that's why they want all the data. They want the granularity. They want everything. They don't want a summary of some haplogroup. They want to know where they are on this huge tree of their lineage. And that, I mean, as we're talking right now, I feel like that sort of genealogical tool is relatively mature and it's scaling. And this is why I think you estimated there was like 1 million kits sold in 2012. I mean, isn't Ancestry DNA?
Well, it was a total of 1 million up to that point. So
that's
it was 1 million over the course of a
cumulative, right?
And then it took maybe 18 months to sell the second millionth.
Yes.
And then it kept scaling
now
beyond
it's
that. And
monthly.
More
than 1 million a month.
Probably across all the companies.
More than 1 million a month now. I mean, that's so the velocity.
Even
is
relatively
so high.
new companies to the game like MyHeritage. I mean, they sold 1 million in the last year.
Yeah.
And they kind of came out of nowhere in that respect.
Yeah, and a lot of those were overseas. And, this is something interesting. With Genographic, I mean, National Geographic, it is called National Geographic, and people assume that it has some government affiliation. In fact, it is non-governmental. It's nothing to do with U.S. government. And many of us argued for a long time internally, like, we should be calling ourselves International Geographic. So we had a very international scope. And we made an effort with the Y chromosome and mitochondrial tests, kind of the phase 1 of the kits that were sold up through 2012, to translate all the materials, the website, everything inside the kit into French and Spanish, and to market those in Europe. And, there was a little bit of interest, but nothing like what you saw in the U.S. In part because, with those relatively blunt tools, we were telling stories about things that happened thousands of years ago, and we could basically say, you're Western European, and the average French person was like, I know I am Western European. What is exciting about that?
Yeah.
So, I think my heritage has sold a lot of kits outside the U.S. But technologically, what's driven the field forward is the price of the technology, which is initially used in research labs and is relatively expensive. So chips that cost hundreds to thousands of dollars come down in price to the point where a consumer kit can be offered for under $100. You can do the analysis. And we get to Helix, which is now going way beyond the chip technology to something called Exome Plus. And Gareth, tell us a little bit about that.
So a quick one-sentence 101 on genomics. Your genome is comprised of regions that code protein called exons, and that make up your phenotypes. These proteins literally build the body that you have. And so when we sequence the coding portion of the genome, we call it the exome. And so that's what Helix is offering, a sequence of all of the coding parts of your genome, and then plus a little bit more to help us do things like our ancestry apps.
The non-junk DNA.
Yes, the non-junk DNA.
Junk DNA.
Yeah, that is
There's
the popular term. Yeah.
Junk DNA is the term that people use for parts of the genome that don't do anything direct.
Or at least we don't know what they're doing.
We don't know. They don't code, as Gareth was saying. They don't code. They don't translate into protein, right? So that's vast. That's 99% of our genome.
But they may have an effect on expression patterns.
Regulation.
Regulation.
Yeah, there's other things structurally, and that's why people do basic science.
Yeah.
There are people who are studying that. But so Helix with its exome is not looking at the junk DNA, except for there are some, it's a plus, there are some marker positions outside of the exome, which, is about 1% of the human genome.
Right.
But so, you're collecting roughly 100 times more genetic data than you get off of the average, genotyping chip, SNP chip.
Yeah.
And so it's
on that.
It's a lot more information, and it's skewed toward the coding regions, and so these are things that should have some sort of functional effect.
Sure.
Yeah. Yeah.
So as we think about, ancestry, and we have, our new regional ancestry product, which, is akin to other things that are available in the marketplace. We're looking at genome-wide patterns of SNP variation among populations and saying, which regional groups are you most closely affiliated with, and so on. But we can also start to talk about, how has your ancestry had an impact on traits? And so a lot of our products are actually making use of the fact that there is this skew toward the coding regions and things that affect proteins and expression levels and function.
Yeah, in the quote-unquote junk, the non-coding regions, there's obviously there are things going on there, but they are relatively subtle.
Sure. Another word that people use to describe it is also like dark matter, because we're not really sure what it does.
The dark genome.
Right.
Yeah. So, focusing on the exome, that's where a lot of your Mendelian diseases are going to be. There are some regulatory ones that are not in the exonic positions, but, if you are causing mutation that has an effect on a protein, that is usually a problem, depending on the protein.
So Helix is able to offer this exome at a sub-$100 price point.
At sub-chip prices.
At sub-chip prices, or comparable.
Comparable, yeah, because the chips themselves are very cheap now.
Which is, again, if you think about what you were talking about, Human Genome Project, $3 billion.
$3 billion.
Over a decade, and now you can do it in a few days for less than $100.
Yeah.
At least get the exome.
Yeah, at least the exome.
What about the whole genome? When is that going to come down in price to, well under $100?
Well, so the most recent things that I am reading is they're targeting about $300 in the year 2020. Mm-hmm. The issue that I would bring up there is these are always linear projections based on current rates of technological change. And I think the reason that genomics has, totally destroyed Moore's law is there have been periodic bursts of innovation, and it's really hard to predict in the next 2 years if, to be explicit, Oxford Nanopore really steps their game up. The whole industry is going to change reactively, and you could see an order of magnitude as opposed to a fold change.
Yeah, Oxford Nanopore is amazing tech. Gareth, do you want to walk us through it? It's a completely different way of sequencing DNA from what people have been using for a long time.
I'm not an Oxford Nanopore engineer, but at the highest level, Lumina Machines, which is the company that they're trying to break into the industry from, they cut your DNA into small fragments, about 200 base pairs or longer. There's some other types of sequencing methods that go on there, paired end sequencing. And then they try and piece together like a jigsaw puzzle to a certain, until that jigsaw puzzle covers you 3 billion base pairs of positions. Oxford Nanopore.
Yeah, well, and so just to clarify, I mean, synthesis, you're basically utilizing the fact that DNA is double-stranded to synthesize the other strand, and because you've got some of these nucleotide bases marked with different fluorophores, different colors,
right?
You can detect what those bases are as they're added to the chain.
Yeah, called DNA sequencing by synthesis.
Sequencing by synthesis, but this is the same method that even goes back to like Sanger sequencing. Effectively,
right.
That's what Sanger is as well. So every method that people have used up until recently to sequence DNA, everything that powered the Human Genome Project was based on this sequencing by synthesis in one form or the other. And then Oxford Nanopore is different.
Right. So Nanopore, as the name suggests, uses very tiny pores to read lengths of DNA as they pass through the pores using electrical current. And as we mentioned, DNA sequencing by synthesis uses light. The Nanopore instrument, probably most remarkably, is that it's the size of a USB drive.
Yeah.
Oh, yeah.
The first time I, so, you know, Gareth said Illumina machine. Illumina is a machine.
Small car.
Yeah, I mean they're like large fridges.
Well, it depends. My Seeks are desktop, but
yeah.
Most
I mean,
of
but
them
these are big metal boxes.
Yeah, the high Seeks.
Yeah.
And
well,
especially
Nanopore. The
Exome.
first time I saw a Nanopore instrument, I knew intellectually how small it was going to be, but I kept staring at the lab bench.
A geneticist from 2010 would never believe it.
No.
No.
It's like because it's like a USB. It's a little USB.
No,
thing.
it is. And in fact, they're working on one that plugs into the bottom of your iPhone.
Yeah.
Oh yeah.
Yeah.
I mean,
So
it's crazy, and the technology again is totally different because what you're doing is you're pulling a single DNA strand through one of these tiny microscopic pores using, an electrical differential in effect. And it's almost like pulling, a beaded necklace through your fist, and each one of the nucleotide bases is a different shaped bead. And by feeling the shapes of those, i.e., how they kind of react as they move through the nanopore, you can read off the sequence and it goes as it slides through. Yeah. That's really crazy.
Yeah.
Yeah.
All right,
cool science.
I mean, this is like serious nerd alert.
Yeah, we can really get
this
into
is like
this
serious
if we want
nerd
to.
alert, like intermission right here. Get some weird looks in the office, but
no, the reason I want to go here is getting back to you, Razib, and this whole issue of whole genome sequencing.
Yeah,
you actually were one of the early adopters.
In a way,
consumer-facing or personal. Genome sequencing.
Yeah, so to focus it on numbers. The first human genome cost about $10 billion, right? Just inflation adjusted, or not 10 billion, 3 billion, 3.5 billion, something like that. So in 2014, as many of you listening will know, but you can Google it, my first son was sequenced whole genome at 10x coverage, which means, it's reasonable quality, but it's not medical grade quality, in utero. Probably.
Why did you do that?
Because I could.
So it was curiosity.
Yeah, it was.
There was no
mostly
medical reason.
No, there was no. There was like one minor medical reason. In terms of like I have a risk for a particular illness, which it's not chronic, but we were curious to see what his would be before he was born. But mostly it was 99% curiosity. And, he was about like 3-4 months in utero. We got the reads back when I think he was 5 or 6 months, and I was analyzing them. So I confirmed that he was R1A1A. So there was, just wanted to double check.
Knew he was yours.
Yeah.
But how did you manage to do this? Because, the average person.
Yeah. I mean, how I managed to do it is, I'm a genomicist, and I know other genomicists, and it was kind of like going back to the cottage industry that you're talking about in terms of someone who knows someone who has a lane that they can make available. And so that's what happened. I got to hook up privately in terms of like researchers who are sequencing a lot of stuff constantly because there's always sequencing going on. The facilities are going around the clock, right? And so I got one of those lanes. We got the genetic material. And we ran it. I got the reads back, and I did the analysis myself. And it was a $3 billion. We're talking like hundreds of dollars, really, in terms of what it cost. Which is like that's 2014. That's about 15 years after the human genome. So we go from 3 billion to a couple of hundred. You know?
And so because this was you decided to share this far and wide. And what was the reaction?
No, I mean, so what happened is when it was done, I had assumed that someone in China had done this. Actually, all of us involved had assumed that I was not the first to have done this ever. And so we asked around, and then I started asking people like Jay Shanduri on Twitter, do you know anyone who's done this before? And I wasn't secretive about it because I just assumed that, oh yeah, there's this Chinese blah, 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 you know? And it turned out nobody knew, and so we were like, okay, that's strange. Then about like 6 months, no, it was a couple of months, I get a contact from a journalist because this journalist, Antonio Regalado at MIT Tech, and we're friends, you know. It was anyway, he would contact Jay every 6 months and ask if any healthy child has been born who was sequenced before they were born, and Jay would be no. And for those of you who don't know, Jay is involved in a lot of this research work, so he would be the one to ask. And then Antonio asked Jay, and Jay was like, actually, I know this guy Razib, and one thing led to another, and Antonio did a profile on the profile was written before my son was born, and I think the article came out a couple of weeks after he was born about his sequencing. So the whole world knew, and it kind of became a little bit of a sensation. It's been translated. The article's been translated to French and Italian, and it's been written up internationally. And, you know, I've had documentarians contact me. So my son's kind of famous. He's not quite clear what that means. And normally he just gives me a funny look and starts screaming at me. But, you know.
But were there people who were like, oh, my God, you shouldn't have done that?
A lot of doctors were extremely angry because I didn't ask for his consent.
His consent is in your son.
As in a 6-month-old fetus.
Like the consent that kids give when their parents vaccinate them?
Yes,
instance?
or what I told Antonio, and he didn't put this in the article because it was edited out because they thought it would be too controversial. What I told Antonio is, well, I can have my son's penis modified when he's born, and I don't ask his consent, and that's not reversible. Or I can look at his genome sequence, which it doesn't do anything to him, right? So Antonio said he actually, I think I can say this now. He said that he actually liked that point, but the editor specifically told him to remove it because a lot of people might be offended because a lot of people have their children or have their sons circumcised without their consent.
Right.
You know.
So that sounds kind of cutting edge.
Yeah.
You know,
2014 cutting edge.
14 certainly, but 2018
a little bit,
somewhat
a little
cutting
bit,
edge.
yeah.
But we're rapidly heading to a time. I think, you know, I've suggested this, you've suggested this. You know, I think Gareth, you probably feel the same way. That within, you know, maybe 5 or possibly 10 years, but I think as recently or as soon as 5 years from now, you know, it might be routine for kids to be sequenced
definitely.
at birth. And how do you guys feel about that? Do you think it's a good thing, bad thing?
I think it's great. I think we need it.
Yeah. What's the problem?
Well, so imagine that world where, you know, the price of sequencing we've talked about in this podcast keeps dropping off a cliff. It's approaching free. Yeah. Suddenly you've got all this data, you know, eventually from tens of millions, even hundreds of millions of people.
Yeah.
And, you know, we're starting to see that in some countries around the world. I mean, the UK Biobank is probably the best example that's kind of publicly available, and lots of geneticists now use that as a resource. Tell us a little bit about that.
Well, Biobank, I mean, that's called socialism, you know? No, I don't normally say good things about socialism, but basically, you know, the British government, they control their nation's healthcare lock, stock, and barrel, and so they can organize from on top, and, you know, they have this biobank, which genotyped, I think it's like 500-some thousand marker positions.
Mm-hmm.
And
with the phenotype data.
With the
which is
phenotype
what makes
data.
it so
with
powerful.
the phenotype data, which in the United States, because of like the lack of organization in our healthcare sector, it's probably not feasible. They have phenotype data. So they're getting all these associations out of these hundreds of thousands. It depends on what trait you're looking at, you know?
And so these would be diseases,
Diseases,
obviously,
yeah.
but also phenotypes like educational attainment. So did you graduate from high school, college?
Yeah. I think they're
graduate
looking
school?
at unibrows, just all sorts of strange things. All sorts of strange things.
So what's the funniest association that's been discovered? Or most interesting? Oddest?
I thought the unibrow was pretty.
Yeah.
Yeah, I mean, that was... How do you record that? I don't understand how the doctor is recording that. I think it probably has
Or
to do
why
with
necessarily.
it might have something to
There's
do with
taste. I think there's even like a traffic citation one. I'll have to double check that. But
there's a lot of
risky behavior.
It's funny, but the reality is we're talking about genomics reading, and then there's CRISPR, which is writing, which is a whole different
and we'll cover that in another podcast.
Yeah, we'll cover that in another podcast. But you have to read everything before you can write. And if we do get all this data, there is the possibility that before this generation passes, we could get rid of most Mendelian diseases. That is not trivial.
And so how would we get rid of them, so to speak?
We could screen. Just
Okay.
do like proactive screening.
So prenatal Mhm.
You could do screening.
Pre-implantation?
Pre-implantation screening. These sorts of things.
So if you're going through in vitro fertilization, yeah.
Just select the egg that doesn't have the trait of interest or not interest. And then obviously, like with CRISPR and genetic modification, if it's in utero or you detect it later, and there's a lot of people walking around with Mendelian diseases, in theory, you could fix some of these people. With genetic engineering. So, I think that is the low-hanging, immediate, obvious fruit that I see before us. But there's other things. Who knows? Until a technology becomes commodity, you don't really understand what people will use it for. So, for example, the spreadsheet. It was not really understood that the spreadsheet would take off the way it did in the 1980s until the spreadsheet was released as an application. You don't know what the killer app is until you release a bunch of apps.
Yeah, absolutely. There's a famous study I remember vaguely. I think it was a McKinsey and Company when they were looking at early adoption of giant handheld cell phones like back in the 80s. And they estimated the total market for this being, a few 100,000 around the world.
Yeah. People did not understand.
Your average herder in Ethiopia is carrying a cell phone now. So.
Yeah. And, it was just like when the iPhone first came out, it was expensive and it didn't have all the security and control features that the BlackBerry did. But it turns out people like using the iPhone personally, so eventually it also came into the business world. The business world initially resisted. So, there are these things where, like, you can have the best laid plans governmentally, institutionally, as a visionary, as a futurist, but you got to have it out there to see what really takes off, and that is what's going to happen, the unknown, which is exciting and probably a little scary.
Yeah, it needs to be made available.
And it will.
Yep.
The prices are coming down. There's no way the prices aren't coming down to commodity levels. And in terms of sequencing, if you have children and you could sequence them, I understand that today some people are scared of genetic technology, but it's going to become ubiquitous. And as a parent, when it comes to your children, you will spend the money. So if it's cheap,
we do anyway.
Yeah.
If you think about if your kid is having trouble in math, you're going to hire a math tutor. You're going to send them to summer enrichment programs. You want them to get into the best school. You're willing to foot the bill for expensive college tuition, et cetera, et cetera. This is a relatively inexpensive, currently relatively non-invasive way of helping them, from a very early age. I think it will be widely accepted. And I think there will be social pressure for people to do it, actually.
Yeah. In countries with socialized medicine, with state medicine, they're going to do the actuarial math and realize that early testing and early intervention,
it's preventative.
It's preventative. It will actually save them a lot of money as opposed to catching something later on.
Well, I'm not a bioethicist, and I don't think we have time to go into the bioethical questions.
Yeah.
And we certainly will revisit those in a podcast in the future. But this does raise a lot of questions about the specter of eugenics and what happens when everybody is evaluated genetically before birth, and how does the population change over time. So that is to be discussed later. We'll leave you guys hanging. But this has been great. Really good overview. Thanks, guys.
All right.
For more information about Insitome, our podcasts, and our genetic products, check out our website at insito.me. That's INSITO.me.